Fantastic. Thank you, Halo. Thank you, Ebony. And Eb's right, we are all volunteers here tonight. And in fact, when Ebony volunteered me to come and speak, I thought to myself, well, what can I tell a room full of people about that's going to be mildly interesting? And so I thought to myself, well, I'll tell you about my career in science, because that's interesting to me. I've had a lot of opportunities. And what I thought I'd do is just show you some of these opportunities that I've had throughout my career. Now, you should be able to notice a common theme in all these pictures. Can you notice what it is? That's right, I'm very happy in all of these pictures. And that's because, as a job, as a career, all careers have a lot of ups and downs, but this one's always made me happy. And in fact, my start in research, this is my favourite toy when I was a child. And you might think to yourself, is this guy just a nerdy scientist? And you'd be correct, yes I am. But despite that, I managed to find myself a lovely wife who's in the audience here somewhere. And I know, I never would have expected it, but it happened. And, we've started a family, and then my son's also got a lot of lovely toys that he likes to play with as well. So there you go. So the love of science has run through my family for a while. Now, research. This is a quote we all live by. To steal ideas from one person is plagiarism. To steal from many is research. And so what I'm going to show you guys tonight is a whole bunch of pictures and videos that I've researched off the internet that will hopefully tell you about my career in science, my journey, and this really fascinating model that we use. And before I get to that model, I should tell you I run a research lab. I do work at LIMS with Ebony and also our third speaker, Patrick. And there are a few scientists here tonight, so I'll try and keep the science to a minimum. If I start boring you, just yell out, Seb, you're boring me. I get that a lot in most scientific talks I get. So what does my lab do? So really what we try and do is understand how all of us go from this point here, which is a single fertilised cell, into this rather cute looking baby here. And the question is, how does that happen? And we always think about what we really try and understand is what happens when this process goes wrong. But what I'm always amazed by is just how often it goes right. And you think, most of the time, it goes completely right. And so what we do is we look at genes. And when we try and understand genes, we try and understand what they do, what happens if they go wrong, and how this impacts this process. Now, normally, anyone who sees genetics in popular media, it's not a topic that comes up too much, unless you get this. Now, I promise that what we do is a lot less controversial. We're not talking about eugenics. We're not trying to create a master race. But just for the sake of argument, we look at genetics and genes with a G. And really, this is how I'm going to start getting to the point, is how genetically similar are we to this incredible little fish that scientists like me use in the lab. So, again, this is some research off the internet that I found. And the question is, how genetically similar are we to other life forms? And so I've got some numbers here. So number one, how genetically similar do you think we are to chimps? Very is correct, 98.8%. So we're basically identical to chimps at the genetic level. What about dogs? What do you think? Am I just going to get very for every question? Throw a number at me. What do you think? 90. Let's have a look. 94. Was that 84? I can't tell. 94. We're 94% similar to dogs. Dogs are lovely creatures. We are quite similar. Most of us are quite lovely. What about cats? Less, correct, 90%. Although probably a lot of people are similar to cats, just if they're assholes, I think. I do realise the irony of wearing a cat shirt while I say that, but yeah, so some people more similar to cats. Then move down a bit to the mighty cow. How similar do you think we are to a cow? 85. 85, almost, 80. 80% similar to a cow. Again, I can name you a few people who are very similar, but I won't. Another organism people use, the fruit fly. What do you think? 78. You're a fly researcher. I hope I got this right. It's 60. Trav will be hearing about this. So we're about 60% similar to a fruit fly. So this is still pretty similar because the next one is a banana. How similar do you think we are to bananas? 60, 40 to 50. So if you think to yourself, next time you go to the fruit aisle, that's like a distant relative you're getting. So, bananas. And so the question there is, where do you think this fella fits in? The zebrafish. 70 to 75 is absolutely correct. You guys know your science. You're getting all the right answers. You weren't the ones to put these slides together for me, were you? Thank you anyway. Yeah, so if you look at this fish here, we're basically 70-75% identical to a fish. And so that's one of the things that makes this such a great model, is because a lot of the things about a fish are the same as what we have as well. And you might think to yourself, and a question we get is, why do scientists use zebrafish and not any other kind of fish? And in fact, I'd never thought about that until I ran these slides past my wife on the weekend, and she said, why do you use zebrafish? And I'm like, I've got no idea. So I actually had to look it up. And apparently this fellow here, George Streisinger, in 1981, published a paper in a really, really highly respected journal, Nature. So the scientists in the room, you'll know what a big achievement this was. And I'm not going to tell you about the science, but basically this fellow did a really cool genetic experiment, and he just happened to use zebrafish because someone had brought those fish to the lab and they had a fish tank, and he's like, oh, I'll use this is cool. And because he did this work for zebrafish, for no reason other than it was available, this model has since become a model of choice. And it's really good because, like I told you, genetics are similar. So that's a starting point. It's got to be something similar. We can't do the same research on bananas. They're small and relatively easy to house. They're tiny fish, really, a couple of centimetres long. They lay thousands of eggs at a time. The eggs, the embryos, they're clear, meaning that we can see these develop under the microscope in real time. And I'll show you a couple of videos about how this happens. And then, really, really cool thing for us, because, scientists don't just like to sit back and watch stuff. We like to interfere. We like to mess around with things. We like to manipulate genetics. And you might think, how do you do that? Well, very simply, here is a little zebrafish embryo. This is the yolk. This is one cell here. And really all we do is we just inject this cell with a really, really tiny needle. And we can inject all sorts of really, really weird shit into there. And we do. And we see what happens. It's all ethical, obviously, but we do experiment a lot. That's why it's called experimentation. If we knew what we were doing, it wouldn't be called experimentation. It'd be called something different. So how do we house these little fish? Basically, in the lab, we have many, many racks like this. And each of these racks here has its own temperature control. It's all ammonia control, temperature, pH, all that. And each of them houses little tiny tanks, all these little tanks. And you can see each of these tanks houses up to about 30 or 40 fish. And then, how do we go from this to getting embryos that we can look at? We take these fish out of here and put them into a little tank. Now, for those of you can see, there's a little divider up here where basically we put the females on one side, the males on the other side, and we kind of get them randy, and then we just leave them there overnight. And so they can see each other. They can't do anything. Until I'm not going to end it there because that would be terrible. But we come in the morning, we take the divider out, and then what happens is that these fish, and if this works, and this isn't working, yes, there, oh no, what happened here? Yeah, it's funny, worked in practice. Okay, it's working here. So hopefully, what these little fish do is they swim up to the shallow end of the tank, and I'm not sure if you can see, coming in here, all these tiny little eggs drop down through the divider. And then we basically just take this out, put these adult fish back here, and we collect all the embryos from there. And now we've got a whole lot of tiny little embryos to do some work with. And the other thing Ebony said to me when she asked me to do this, she said, Seb, whatever you do, don't just pull out a whole bunch of slides for your scientific talk and put them into this one. And so, of course, the first thing I did was pull out a whole bunch of slides from my scientific talk and put them into this one. But This is a slide that I usually start with, and I like to show this video for how a zebrafish embryo develops. And so here is the yolk again, and up here, this is time-lapse microscopy of a zebrafish embryo developing in real time. And I won't talk you through all the complex processes. There's a whole lot of research that's gone into that. But what I really wanted to show you is this slide here. And so whenever I give a scientific talk, and I should say most of my talks are scientific talks, and I don't often get a chance to talk to real people, so thank you all for actually giving me this opportunity. But I like to start with this, because to just show you how similar all these organisms are at an early age. So fish, turtle, chicken, pig, human. If you look at them at this stage here to what this embryo is now, these things are basically identical. So the earliest stages of how we go from a single egg to an embryo are identical. Now obviously after a point we have differences, but the other thing I'd like to point out, and this will become relevant later, is that all these species, they've got a very definable head and a face. And as I'll tell you in a little bit, one of the major birth defects we look at are problems with the craniofacial skeleton. And so these fish have a mouth, they've got a head, they've got a jaw, they've got eyes, they've got a brain, and those things do exactly the same things in a fish as they do for all of us. And so the question I'm going to ask you here is, how long do you think it's taken this little fish here to get to this point? 5 days, a week? It's less. It's more than 6 hours. 24 hours is absolutely correct. So in 24 hours, we go from the tiny little egg to this. And in fact, this is what the embryo looks like. Now, I told you before, you can see through it, which you can. If you look here, you can see the eye. You can see into the brain. This is the brain of the fish right here. You can see it. This thing here is the ear. These things here are the developing muscles. And so even before we realise how useful this is, I just reckon this is bloody cool. You can look at an animal and see its organs. And of course, scientists aren't happy with just looking down a microscope. They want to do movies. They want to do images. And so here is a heart beating. You can see the heart beating in real time. Here, hopefully, you can see the blood cells, individual blood cells moving through the blood vessel. So you can just take your garden variety microscope, like the one I had as a kid, and you can put an embryo under it and you will see this happening. Now, that's not cool. I don't know what is. But it gets cooler. It gets actually a lot cooler than that, because that's cool enough. Okay, so you can see blood moving, you can see the heart pumping, magnificent. Then, this is the cool stuff. This is the cool part of the talk. So if you remember nothing else, just remember these cool things that I'll show you. So ages back, other researchers knew that this fella here, this jellyfish, if you put it under a particular light, it would actually glow green. And it glows green because this fish has a protein called green fluorescent protein. Scientists aren't always an imaginative bunch. But, see, you won't forget that. Now you know what it does. It's a protein, it's green. And I'm denied about giving you a history lesson, and I thought, you don't want a history lesson, so I'm just going to run it shortly. But this was discovered in 1961. Then in 1994, people discovered that you could actually cut this gene out of the jellyfish and start sticking it into other animals. And so, of course, what happened to those animals? They glowed green as well. Now that is really starting to get to the Sydney Sweeney side of genetics. But we do it for good, I promise. So in 1988, and this is one mainly for my friend Steve Petrovsky, who's given a talk before. But in 1998, another researcher discovered that if you tweak that protein, you can actually make all sorts of different coloured tags. And this is, for those of you that have ever done university microbiology, this is a bacterial plate, and bacteria expressing all these different fluorescent tags have been streaked out onto that plate and it's made this beautiful beach scene. Oops. And in 2008, the Nobel Prize people saw fit to award this discovery the Nobel Prize for Chemistry. And so what does this fluorescent tag do? It basically allows us to look at individual organs and cells in fish. Here, what organs do you think this is? Blood vessels is absolutely correct. I know you said muscle, but I'm telling you blood vessels. So you can actually stick that tag onto a protein that's just expressed in blood vessels, and you shine a light on the fish, and you can see all the blood vessels in that fish in beautiful detail. What about this one here? This is a top-down view. What do you think? Brain. That is the brain, and you can see just how beautifully this entire brain lights up green. You can even do cool things like make gonads. So testes in male light up green, the ovaries in female light up red, and so you can quickly tell which ones are the males, which ones are the females. And then some other cool techniques, and I won't tell you about this one in any sort of detail, but one other thing researchers do with the fish is you can actually transplant other cells into a fish. And this is actually a tumour. This is a cancer that's been put into a fish. And so you can see the cancer in purple, and then the cool thing you can do is see what effect does that have on the blood vessels. And so, of course, you would see, for example, here there's a lot of those blood vessels, and here you can see a lot of them have been destroyed, and that's because of the cancer. So you can actually see this disease happening in real time. And I wanted to show you guys these movies because here we combine the time-lapse microscopy with the fluorescence, and we can see things developing in real time. So up here, this is some muscle. This is time-lapse happening over time. Up here on the right, I think this one's really cool. You can actually watch individual cells. This is the nervous system, again, developing in real time, and you can just see how beautiful that whole network of cells forming is. Over here in the bottom, oops, sorry about that. Best laid plans. There's a big reveal that's just gone. Well done. I'm not blaming you, Ebony, but this is all your fault. No, I think I'm okay. Let's see. Let's try that again. Where was I? Just chat amongst yourselves. Okay, here we go. So I was going to show you this one, which is blood vessels not developing in real time like they should. There we go. This is the blood vessels forming in real time, and you can see again individual cells happening. And then the last one is, you might remember a few slides ago I showed you a beating heart, which is cool enough, but here you can actually see the heart is green and the blood cells are red. And so you can actually watch it happening, you can record it, you can measure heart rate, you can do blood pressure tests, you can do all sorts of cool stuff because you can see that happening in real time. And so just in the last couple of slides, I thought, well, I've shown you all this other research from all the other people, but do I actually have any credibility telling you all this? Look, what has my lab actually done? And I do notice a couple of lovely students in my lab, April and Jared here in the front row. Say hi, guys. Hey, they love this. Yeah, give them a round of applause. There they are. This is a lot of their work, so I'll give them the credit they deserve because I just stand up here and talk about it. These guys do the work. But what we do, I told you we work on craniofacial defects, craniofacial development. And for example, here is just one fairly typical defect. It's called micrognathia, or basically having a small lower jaw. And this has led me to partner with this wonderful charity who I just wanted to give a big shout out to tonight, let all of you know about the fantastic work they do in supporting patients that have these craniofacial defects. And I like to support them because they support me with giving me research money to do this. And one of the things I had the pleasure of doing recently was I attended a panel, and I got to meet this lovely young lady, Scarlett, who, you know, very smart, intelligent. She's about 10. And then I had a panel, and I was sitting there in front of like 60 people, And this little girl asked me a question, and I'm like, I've got no idea. So she asked me how many children get born with the same craniofacial defect that she has, and I'm like, I don't fucking know. But I didn't say that because I sort of managed to sort of come up with an answer, and I said, there's about 3,500 to 4,000 children every year in Australia that have some sort of difference in their face. So it's actually quite a significant problem, and there's not enough funding. So if any of you want to make a donation to Craniofacial Australia, just please do so. That's a little plug. And so the last sciencey thing that I'm going to tell you about is once we know what the problems are in humans, we can make the same problems in fish. And this other technique called CRISPR, which basically allows us to cut DNA very specifically with a pair of scissors almost, and then it gets stitched back together, but it gets stitched back together wrongly. The fish can't repair it correctly. And so what you can end up happening is you actually get fish that very nicely model the same problem that these patients have. And so you can clearly see here is a jaw in the fish, and here is a defective one. Excellent. And so what I just want to show you, a couple of slides, is how can we use the fish to understand craniofacial development. This is another microscopy photo of a very young embryo, and you can see the mouth forming here, the nostrils, and the face. Again, this is the photo I just showed you. Faulty gene versus normal gene. You can very quickly see the differences. And another thing we do is we can actually look at the cartilage and the bones of the face. We just put a dye in, and here, normal, here, faulty. And you don't need to be a scientist to know that that looks different to that. At least I'm hoping that's the case. This one, everything's right. This one has a lot less of those elements, which explains why they haven't got a jaw. But we can go even deeper, and this is some of April's work here, where we can look right down at the single cells that make up that face. And here, again, all these hundreds of thousands of normal cells, and here you can see a lot of those cells are missing. And so this fish allows us to discover what are the consequences of those genetic defects. And I think I just have one more thing, or do I? Oh, yeah, you know, all right. Are you going to bear with me for one more video? Hopefully. All right. Well, it's actually two, but it's one. I'm going to hope this works. One cool thing that we're doing at the moment is that we have these fish with a faulty gene. And what we discovered is that, keep your eye on the three that have a faulty gene. This is normal development here. Faulty, faulty, faulty. So it's developing. This is the same as the movie I showed you before. And keep an eye to see what happens. They explode. Why do they explode? I don't know, but we're working on it. But one thing we have done is we changed their environment. We put these into different water in a different facility. And all of a sudden, what happened now is here we've got two normal, two faulty. Oh, that was good. Well done. Wrong button. See, just making sure you're paying attention. You still with me? Okay. Normal, normal, two faulty ones. You can see the faulty ones don't look quite right, but they don't explode. And so they actually get through this explosion stage. And all we did was put them into different water, into a different environment. And so what this means, we've got a whole brand new model to test the effects of environment on development. And so what we've done is a cool experiment. I feel like I'm using the word cool a lot. I'll say neat. How's neat? A neat experiment. We took our fish, adults, we bred them at La Trobe Uni. We collected these embryos, we split them up into three. We took 70 and put them into 3 different facilities around Victoria. And because I don't want to get sued, I'm going to call them Facility 1, 2 and 3. If anyone wants to know, don't ask me on stage, but I'll happily tell you where they are afterwards in private using a false name. And so we've done a really deep dive into trying to understand just how much effect does environment have on development of a genetically sensitized fish. And because I'm a scientist, contractually I'm obligated to show you a graph. It's important. What does it mean? I'm glad you asked. All we did is we looked to see how well do those fish survive at each of our 3 facilities. And you can see here, facility 1 and facility 3, they're basically the same. They survive just fine. Facility 2, they're buggered. That's really not good for them. And they end up looking like this, which again, you can tell that's not what a fish, by now you can tell that's not what a fish embryo looks like. So, what's causing this? That's still top secret, which is again scientist code for I have no idea. But that's not quite true, because we have some idea. We know that it involves heavy metals in the water, and we know that these metals are driving a change to the bacteria in the gut, or the gut microbiome. And the really beautiful thing about this as a model for human development is that you probably all know, if you've ever been pregnant or had known people who have been pregnant, you want to avoid heavy metals and you want to avoid viruses and bacteria during pregnancy, because that can impact your health of the baby as well. So here we've got another link between fish and humans. It's not just the genetics, but it's the environment as well. And so basically that's my life, and I thought to myself, I've actually got no idea how to end this talk. So I put into ChatGPT and said, ChatGPT, how am I going to end this talk? And ChatGPT comes up with this exact quote, which I've pretty much copied. We spend our days making genetic typos in tiny transparent fish, shining expensive lasers at them, and trying to work out why some faces and brains don't build themselves quite right. And I couldn't summarise that any better myself. And so I just want to leave you with, in my career, I've gone from being this young, happy scientist here to now running a wonderful lab with, this is a reasonably recent photo, and you can just see all of us here with our glasses of happiness as well, doing our work. And the last thing I wanted to do is, you might think, why is this here? Because in addition to being a nerdy scientist, I also run a trivia night company called Trivia Buffs. And I thought I'd just give myself a free plug in a room full of people who I guess like trivia. So that's it from me. Thank you.
You know, I had no idea that we had a trivia buff in the room when I put together my ragtag trivia.
Well, I was competing at the start. I know you said to be backstage while the trivia was on, but I'm not missing this.
Oh, Jesus. All right, we have time for a few questions for Seb. If you just want to raise your hand. Okay, one at the very back here. Oh, I'll just make sure everyone heard that. So what's one of the biggest and coolest discoveries that have come from zebrafish as a model? Is that right?
Well, I think I've just shown you all of them. I think probably the biggest thing is that there's been a couple of studies looking at the circulation and the blood system where some human diseases have actually been completely reproduced in the fish. And then the beautiful thing is that that fish was then used to test drugs and medicines. And so I haven't spoken about that side of things, but obviously the fish living in water, you can put any sort of medicine or drug or anything into that water, and you can see, is it going to work to cure the disease? And one of the biggest impacts the fish can make is when you can directly find cures that go straight into the clinic and can cure human patients as well. So I'd probably say that, in addition to all the cool stuff.
Yes, one up here. So the question was a twofer. You said you test the blood pressure of the fish. So what was it?
I was hoping no one's going to ask me that because I've actually got no idea. I think you can test the blood pressure. But I don't know how or what it is. So you could have literally asked me about anything else I said. And I could have answered that one. But no, I don't know. But it's lower than humans.
And the second part of your question, sorry, I've already forgotten a bit of it, but you were comparing... Oh, in Rosanna. That's what... Okay. So there's high diagnoses of MS or Parkinson's, a neurodegenerative disorder in Rosanna, the suburb, and people think it's to do with the water pipes.
Well, that's really interesting because my sister-in-law and my father-in-law happen to live in Rosanna, and I used to live in Rosanna as well. First thing I'll be telling them about that. Well, it's interesting. I mean, the thing is, so certainly heavy metal contamination is very strongly linked to those neurodegenerative diseases as well. As to whether it's the pipes, that's really interesting. But certainly copper, we need copper, we need zinc, we need magnesium, but having too much of it is bad at the start of life, but it's also quite bad at the end of life. So thank you for letting me know about those filthy water pipes in Rosanna.
That might have been a sneaky way to ask, is that where facility number 2 was?
No, but I'm thinking I need to set one up there and see what happens.
Any other questions? Yep, right here.
Do you plan to combine your love of zebrafish with the knowledge of liquid?
I would say that I have done that very effectively throughout my entire career. Thank you to the free drinks, by the way, Halo and Ebony. No, so actually, one of the other things I was considering telling you about is craft beer and home brewing, because as luck would have it, I am a home brewer and a craft beer lover as well. So I actually find, I don't know about any other researchers in the room, but I always find that I do my best thinking and my best writing and my best marking of student work when I've had a glass or two of beer or wine. So I actually think these things go hand in hand. So, yes, good question.
Up the back, where? Oh, right here, sorry.
You mentioned that they tied it back to the gut biome was the transmission vector for this. How did you get to that conclusion out of all the different systems?
So the question is, how'd you figure out that the gut microbiome was affecting those fish embryos?
Well, that's an excellent question, and that's partly why I can't give you the full answer as yet, because we haven't quite nailed it. But what we have found is that we basically did a whole analysis of the water at Facility 2. So the facility had the worst outcome. We analysed everything. And then we saw pretty much everything is within normal ranges, except copper. It's really high. And at the same time, because I'm friends with a lovely bacteriologist at La Trobe, Steve, he has a whole lot of tools for analysing bacteria, and he said, why don't you look at the bacteria? And so we looked at the bacteria independently of the water. So we're looking at two separate things. But interestingly, what we found was that the bacteria in that facility have evolved over time to be copper resistant. And so those fish, so the fish that have been at that facility for ages, they're resistant, they're fine, because they've got these copper-resistant bacteria. But our fish that we brought over there had no idea what hit them, because they didn't have that bacteria. Even after being there for many, many months, they hadn't colonised their gut with that bacteria. And so I think that's really exciting. It's something we're investigating. It's something we would have been investigating a lot more strongly had our recent NHMRC grant application got up, which it missed out by not much. Not that I'm bitter, but if there's any reviewers here who had my grant, please leave the room now. But yes, no, we are working on it, and we're hoping to really try and nail that down in more detail. It's a great question.
Alrighty, do we have any more? Ebony, can you be my second set of eyes here? Over here.
Hey, so how do we make sure that you talk about how some zebrafish have copper superpowers or something? How do we make sure that the supply is like
So asking how we make superpowered mutant fish in a nutshell. Is that you, Wade, by the way? It is.
Yeah.
Hey. So excellent question. So that's actually a really, really good question. And that's actually one of the kind of assumed fallacies that we have in research, is that every environment is the same. And for the zebrafish, you saw I put up the picture of the fancy racks, and we have, all climate controlled, all temperature controlled, everything's perfect. What isn't controlled is the water actually going into that system in the first place. And that comes just from our water system, or it comes from the pipes in the facility. It's the outside water. So yes, it gets treated, but then if it has too much copper or too much ammonia or too much calcium, that stuff stays in there. And so one of the things that we always, and this isn't us just knowing that different models, different animals have different outcomes in different facilities. A lot of people have known this. But what people haven't done is try and actually attack systematically why. And so I think what our work has done, I'm hoping, is that we've now got a set of fish that we can put into any facility in the world, and they can test in a living organism how good and how clean their water supply is. So if anyone's got a business that wants to commercialise that, come and see me afterwards.
You're such a businessman, plugging your trivia buffs and your fish.
Oh, I've got heaps more.
craft beer, I'm sure, too. All right, maybe one last question for Seb, if we have one. Yes, all the way at the back here. Where'd you get the shirt?
The cat shirt. Well, my lovely wife got that for me, actually, off the internet, much like most of my research, actually. So you'll have to ask her. But I love it, and I thought, well, yes, suits my personality. Not that I'm an asshole, but it just suits my, yeah.
All right, put your hands together for Seb.